Good afternoon, students. Now, today we will deal with reproduction in Equisetum. Now, Equisetum reproduces vegetatively and by means of spores. Now, first of all, we will deal with the vegetative reproduction. Now, here the septarian rhizomes of some species, for example, Equisetum telmetia and Equisetum arvens, form tubers which on separation from the parent plant germinate to produce new sporophytic plants. That means the rhizomes which are under beneath the soils, they will form tubers, and from these tubers, after separation from the parent plant, we can get a new plant. The tubers develops due to the irregular growth of some buds at the nodes of the rhizomes. Now, second type by means of spores. That is, here the reproduction takes place by means of spores. The spores are produced within the sporangia. The sporangia are born on the sporangiophores. That means these are the stalk of spores which bears spores and hence called as sporangiospores, which are aggregated into a compact structure from as strobilus or cone or sporangiferous spike also. Now strobilus. Now here strobilus, it is a cone-like structure. The strobilus is terminal in position and generally is born terminally on the chlorophyllous vegetative shoot. However, they may be born terminally on a strictly non-chlorophyllous axis also. For example, Equisetum arvens. The the strobilus is composed of an axis with whorls of sporangiophores. Each sporangiophore is a stalk structure bearing a hexagonal pelted disc at the distal end. On the undersurface of the sporangiophore disc, 5 to 10 elongate cylindrical hanging sporangia are born near the periphery in a ring. The flattened tips of the sporangiophores fit closely together, which provide protection to the developing sporangia. The axis bears a ring-like outgrowth, so-called annulus, immediately below the walls of sporangiophores, which provide additional protection during the early development of spore. Now we will see the development of sporangium. The mode of development of sporangium is eusporangiate. Now you will see that you will keep in mind that the development of sporangia is here eusporangiate type. That means here sporangium develops from a group of cells rather than the single cell and with no specialized dispersal of spores. Hence such type of development, it is called as eusporangiate type of development. Here the superficial cells adjacent to the original initial may also take part in the development of sporangium. Sporangia are initiated in a single superficial cell around the rim of the young sporangiophore. The periclinal division of the sporangium initial forms an inner and outer cell. The inner cell by further divisions in various planes give rise to the sporogenous tissue. The outer cell by periclinal and anticlinal divisions give rise to the irregular tiers of the cells, the inner tier of which may transform into sporogenous tissue and the outer tier becomes the future sporangial wall cell. Now here you can see the diagrams, and here the strobili or cone bears the numerous sporangia in the sporangium. The innermost layer of the sporangial wall differentiates as the tapetum. The sporogenous cells separate from each other, round off, and eventually transform into a spore mother cell. All but the two outermost wall layers disorganize to form a periplasmodial fluid. However, not all the sporogenous cells function as spore mother cell. Many of them degenerate to form multinucleate nourishing substance for the spore mother cell. Each spore mother cell undergoes meiotic division which is a reductional division and produces spore tetrad. All spores in the sporangium are of same size and shape, that is homosporous. Now the mature sporangium. We will see the structure of mature sporangium. The mature sporangium is an elongated sac-like structure attached to the inner side of the peltate disc of the sporangiophore. It is surrounded by a jacket layer which is composed of two layers of cells. The inner layer is generally compressed and the cells of the outer layer have helical thickenings which are involved in the sporangial dehiscence. That means here the thickenings, they are special type, they are called as helical thickening zigzag, and they are called as helical thickenings, and they are involved or they are helpful for the dehiscence of spores. Now dehiscence of sporangium. At maturity, the strobilar axis elongates as a result of sporangiophores become separated and exposed. The sporangium splits open by a longitudinal line due to the differential hygroscopic response of the wall cells. Now the spores. Now we get the spores which are liberated through the sporangium. Now the spores, they are spherical and filled with densely packed chloroplast. The spore wall is laminated and shows four concentrate layers. The innermost is the delicate intine followed by thick exine and a middle circular layer and the outermost epispore or perispore. The intine, that is endospore, and the exine, that is the exospores, are the true walls of the spores. The outer two layers, that is cuticular layer and epispore, are derived due to the disintegration of the non-functional spore mother cells and tapetal cells. At maturity, the epispore, that is the outermost layer, splits to produce four ribbon-like bands or strips with flat spoon-like tips. These bands are free from the spore wall except for a common point of attachment and remain tightly coiled around the spore wall until the sporangium is fully mature, and these are called as elaters. The elaters are hygroscopic in nature. The spore remains moist at early stages of development, thus the elaters are spirally coiled round the spore. The spores dry out at maturity and consequently the elaters become uncoiled. Now we will see the functions of elaters. An elater is a cell or, or a structure that is hygroscopic and therefore will change shape in response to changes in moisture in the environment. Elaters come in variety of forms, but they are always associated with plant spores. A plant structure functioning in the dispersal of spores, such as one of the elongated filaments among the spores in the capsule of a leavered bud. A tiny elongated structure that forces the dispersal of spores by an absorption of moisture. It is either a band attached to the spores, as in hostels, or a filament occurring among the spores, as in leavered buds. Now here you can see the diagram of elaters with spores. Now you can see here the elaters, they completely coiled a spore.